Nowadays, there's so many shiny new technologies we can use to study DNA, from Sanger sequencing to PCR to southern blotting, but some of the earliest experiments that really opened up our understanding of genetics were quite simple and elegant. Back in the 1950s, two scientists, Matthew Meselson and Franklin Stahl, were committed to cracking the mechanism of genetic inheritance through DNA replication. One possibility was a conservative mechanism, by which the entire DNA molecule is completely duplicated to create an entirely new DNA molecule, thus conserving the entire original molecule. Another possibility was the dispersive mechanism, under which the DNA backbone would be broken in multiple places as the two strands unwind, and both the old and the new DNA molecules would have sections of old and new DNA. The final possibility they considered was the semi-conservative method, by which the two DNA strands separate, and each serves as a template for the synthesis of a new strand. Under this method, both the old and new DNA molecules would each contain one old and one new strand. To solve this mystery, Meselson and Stahl conducted an experiment where E. coli bacteria were grown in medium containing the nitrogen-15 isotope, which is heavier than the much more common nitrogen-14 isotope. After several generations, this heavier nitrogen isotope had been nearly completely incorporated into the bacterial DNA. Then the bacteria were transferred to medium containing the common nitrogen-14 isotope and allowed to replicate, and their DNA was then extracted periodically and centrifuged on a salt gradient to determine its density. If the DNA had been replicated via a conservative mechanism, after one round of replication, we could have expected to see two separate bands, one heavier for the old nitrogen-15 containing DNA, and one lighter for the newly synthesized nitrogen-14 containing DNA. But instead, all of the new DNA molecules had an intermediate density, suggesting the new DNA molecules contain parts of the old and new DNA. This would fit with either the semi-conservative or dispersive replication mechanisms. So then the bacteria were allowed to continue to replicate, and the DNA was extracted again after additional replication. After 2 rounds of replication, 2 equal bands appeared in the centrifuge tube, one at the intermediate density and one at the lighter density that would be expected for DNA containing just the nitrogen-14 isotope. This rules out the dispersive mechanism of replication, as if this were the case, we'd expect all new DNA molecules to contain some old nitrogen-15 containing DNA and some newer nitrogen-14 containing DNA. Rather, the best explanation was that with each round of replication, the 2 strands of DNA were separated and used as a template upon which a new strand could be synthesized. Thus, the Meselson-Stahl experiment showed that semi-conservative replication best explains how our DNA is replicated, where one strand of the parent DNA molecule is conserved in every new DNA molecule. Fast forward a few decades, and now we have a much more complete picture of the mechanisms governing DNA replication. It's incredible to me that when we think about bacteria, you'd expect that we humans would have very little in common with them. But it turns out that we share a lot of very similar, highly conserved molecular processes, at least to some degree. This is partly true when it comes to DNA replication. I mean, no reason to reinvent the wheel, right? And bacteria are actually a great model for understanding some very basic processes. But there are also some key differences between bacterial and eukaryotic replication. For example, bacteria have one origin of replication, which is a specific sequence that's recognized by what's called the pre-replication complex, and then replication proceeds in both directions along the circular bacterial chromosome. On the other hand, eukaryotes have much more complex genomes, and accordingly multiple origins of replication, and DNA replication produces an identical sister chromatid that's connected to the original DNA molecule at the centromere of a chromosome. A suite of enzymes make DNA replication possible. The first problem that has to be solved is the unwinding of the double helix, since the double-stranded DNA needs to separate if each strand is to be used as a template for new DNA. This unwinding of the helix is accomplished by the appropriately named helicase enzyme, which unwinds the 2 strands of DNA at the replication fork. Now, if you've ever tried to separate 2 intertwined pieces of string, you know how hard it is to keep the 2 strands from rejoining. So our cells came up with a molecular solution in the form of single-stranded DNA binding proteins, which are an army of proteins that latch on to the newly separated strands and prevent them from coming back together. Now, if you were to take a pair of intertwined strings and pull them apart, the strings would become overwound or supercoiled upstream, generating a lot of tension. Our DNA is the same way. In comes the topoisomerase enzyme, also known as DNA gyrase, which alleviates supercoiling created by helicase by making incisions in one of the DNA strands, and then rotating the cut strand around the other and rejoining them to relax the DNA. Then something has to do the initial work of recognizing a start sequence and actually initiating DNA replication, which is harder than it looks, and that's done by primase. Primase synthesizes a short RNA primer that has a free 3' hydroxyl group that's used as a starting point for the synthesis of a new strand. Let's pause to recognize the significance of this. The replication machinery can't just lay down a few DNA nucleotides and go to town. Rather, that 2' hydroxyl group that's only present on RNA but not on DNA is necessary before we can even lay down the first building blocks of DNA. Interestingly, this dependence on RNA is thought to support the idea that long ago, RNA may have been the original form of genetic material before DNA was even cool. So there's a lot of pre-op work that has to be done before we can actually get to DNA synthesis. That synthesis is performed by the enzyme DNA polymerase, rather a number of different DNA polymerases. There are multiple different DNA polymerases in both prokaryotes and eukaryotes. But let's hold this thought until we talk about the basic functions of DNA polymerase. This enzyme travels along one of the separated DNA strands and adds nucleotides sequentially after the RNA primer in the 5' to 3' direction. Since the strands of the DNA double helix run in anti-parallel directions, DNA polymerase must read DNA in the 3' to 5' direction, while synthesizing the new complementary strand in the 5' to 3' direction. So say we've got this replication fork. On this one strand, DNA polymerase travels along the template strand in the 3' to 5' direction, in the direction of helicase, and it's able to synthesize DNA continuously. This is called the leading strand. Simple enough, right? Well, let's take a look at the other strand. Here, reading in the 3' to 5' direction means that DNA polymerase is constantly moving away from helicase, meaning that it's always going to miss the new DNA that's exposed behind it. The solution here is for a new DNA polymerase to latch on as more DNA is exposed and catch up to the rest of the new strand. And then as the replication fork opens up further, another DNA polymerase latches on and synthesizes another DNA fragment, and so forth. This strand is called the lagging strand, and these fragments are known as Okazaki fragments. So the system appears to work well enough, except eventually these fragments are going to have to be joined together. This is performed by DNA ligase, which ligates together Okazaki fragments on the lagging strand, forming phosphodiester bonds along that sugar-phosphate backbone between adjacent nucleotides. DNA ligase also comes in handy when the RNA primer is eventually removed and replaced with DNA nucleotides, which are then joined with the existing adjacent nucleotides. DNA polymerase arguably steals the show when it comes to DNA replication, and as we mentioned earlier, there are actually multiple types. The key polymerase enzymes used by prokaryotes are DNA polymerases 1, 2, and 3. DNA polymerase 1 is responsible for removing the RNA primer and filling in the gap that's left by that primer, and it's also involved in DNA repair. DNA polymerase 2 is principally involved in DNA repair, and DNA polymerase 3 is the main enzyme that's involved in run-of-the-mill DNA synthesis in prokaryotes. DNA polymerase 3 is also capable of proofreading, meaning that not only can it synthesize new DNA in the 5' to 3' direction, but it can also temporarily move in reverse from 3' to 5' to check its work and look for any mismatched bases, which it then excises. This is referred to as 3' to 5' exonuclease activity. In fact, DNA polymerase 1, remember the one that replaces the RNA primer with DNA, it's actually capable of proofreading in both the 3' to 5' and the 5' to 3' directions. Since Roman numerals are already taken, eukaryotic DNA polymerases were named using Greek symbols, but You don't have to know much about individual eukaryotic polymerases. Here's a quick summary of what you should understand. DNA polymerase alpha initiates DNA synthesis, and then DNA polymerase delta takes over, which is capable of much more efficient DNA synthesis. And this polymerase also fills in the gap when the RNA primer is removed. DNA polymerase epsilon helps extend the leading strand and is involved in DNA repair, as is DNA polymerase beta. Finally, DNA polymerase gamma is responsible for replicating mitochondrial DNA. There are a few other types of DNA polymerases that have their own names. For example, reverse transcriptase is a DNA polymerase that synthesizes DNA from an RNA template. This is used by retroviruses like HIV to convert genetic material, which exists as RNA, into DNA so that it can be integrated into the genome of a host. Another special polymerase you should know is telomerase, which extends telomeres, repetitive sequences at the ends of eukaryotic chromosomes, since regular old DNA polymerase can't faithfully replicate chromosome ends. So otherwise the ends of chromosomes would get shorter and shorter over time in dividing cells. This is actually thought to play a role in the aging process. Telomerase helps by rebuilding telomere sequences, particularly in stem cells and cancer cells, but not in most somatic cells. If you think about it, the presence of telomerase in cancer cells tells us that limits in cell division are not necessarily a bad thing. It's not like we learned about all the complex mechanisms involved in DNA replication overnight. We started off with simple, elegant experiments like the ones conducted by Messelson and Stahl, and gradually built our knowledge of how DNA is replicated every time a cell divides, which happens every day in the human body. It's happening right now, even as we speak. And I have to admit, considering the fact that our replication machinery has to faithfully copy something like 6 billion nucleotides in the average diploid cell, it does a pretty good job.